Welcome, everyone, and thank you for joining us today for Know Your NETs, the Net Research Foundation's 2024 Virtual Patient and Family Education Conference. I'm Elyse Gellerman, CEO at NETRF. Our theme today is Hope in Action. Hope is both a noun and a verb. Hope is an optimistic state of mind. To hope is to look forward with desire and reasonable confidence. With neuroendocrine cancer, hope may mean the promise of research and the development of new treatments. It could mean the ability to manage symptoms and live a full and happy life. We want you to gain knowledge and hope from what you hear today from NET specialists and people with neuroendocrine cancer. We're excited that so many of you have chosen to spend part of the day with us. Registrants for this conference represent just about all 50 states and 15 countries. 58% of you told us this is your first neuroendocrine cancer education program. 36% are recently diagnosed in the past 1 to 2 years. We are especially glad you are joining us. If you have not yet watched NETs and NECs 101 by Dr. Aman Chauhan, you can view his presentation on our YouTube channel. I hope you've all checked out your virtual program booklet. You can find the link on the conference landing page or in the email we sent to you a few days ago. This program would not be possible without our co-sponsor, University of Chicago Medicine, as well as Novartis, RayzeBio, and Crinetics. Thank you to all of our sponsors. My co-chair for Know Your NETs is Dr. Xavier Koitkin of the University of Chicago Medicine. Dr. Koitkin is a surgical oncologist with extensive expertise treating neuroendocrine tumors. As the director of the University of Chicago Neuroendocrine Tumor Center, Dr. Koitkin leads a multidisciplinary NET team. Dr. Koitkin and his team have been longtime collaborators with NETRF, and we are always so grateful for their work on this conference. You will hear from Dr. Koitkin later in this program. So we start today with hope in action through research, from basic research through clinical trials. Our first speakers are Dr. Anna Greene and Dr. Don Quelle. Dr. Greene is NETRF's Director of Research. She leads our scientific programs and earned her PhD from Dartmouth College. Dr. Quelle is a co-chair of NETRF's Board of Scientific Advisors. She's a professor of neuroscience and pharmacology and pathology at the University of Iowa Carver College of Medicine. They will discuss the importance of laboratory research.
Hi, my name is Anna Greene, and I am the Director of Research at the Neuroendocrine Tumor Research Foundation. I'm happy to be here with you today to talk about NETRF and how we fund research so that we may have discovery in NETs through basic and translational research. NETRF has a core mission to support patients, families, and caregivers affected by neuroendocrine cancer. But in parallel to that, we also have another core mission to fund research to discover cures and more effective treatments for neuroendocrine cancer. NETRF is a leading global funder of neuroendocrine cancer research because we know this is how we're going to learn more about this disease so that we can learn how it starts, how it grows, and how we can better treat it. But why do we think research is important? Why do we think this is going to help patients? Because we've seen this work. So if we look at overall survival rates and how they've changed over the years, we can see great improvements. So if we look at the year of diagnosis of 2000 to 2004, we see that the 3-year overall survival for NET patients was about 50%. But moving to 2015 to 2018, we see that the overall survival increased to 85%. And so this is a significant increase, and it's attributed to different things, including improved diagnosis, improved classification of tumors, but also improved treatments. And so if we look at the right, we can see all of the NET drug approvals that have happened over time. And most of them have happened after the year 2010. And that correlates pretty well with improvements in overall survival. And so that's how we know that research works, and that's how we'll get to improved treatments for patients. So what are some of the NETRF contributions to basic and translational research? We're really proud of the research we've funded over the years, and some of the findings have been new mutations in nets that have never been seen before, such as DAX and ATRX and PNETs. We've also funded work that's found new subtypes of nets, such as a new lung net subtype, the supracarcinoids. We've funded work that's led to next-generation radioligand therapy using isotopes actinium and terbium. We've funded work that's led to new net models that allows scientists to better study this disease in the lab. We funded work that's validated new targets, such as UPAR, which is heading to clinical trial. And this is important because somatostatin is the common receptor that's targeted, and some patients don't have somatostatin receptors or express them heterogeneously. We're very excited to say that we've also funded work that's led to the first CAR T-cell therapy moving into the clinic this summer. But we still think there's more to come through research. We're very excited about areas like early detection. So how can we use biomarkers to detect the disease earlier so that we could perhaps cure it through surgery alone? We're excited about precision medicine. So how can we integrate molecular and genetic data about patients and their tumors to individualize treatment plans? Immunotherapies. These typically haven't worked as well for neuroendocrine cancer patients. But through research, how can we figure out how to harness the immune system to work against these cancers? Radioligand therapies. How can we improve PRRT to go beyond stabilizing disease to causing major regressions? And so we'll get there through research. And NETRF primarily funds basic and translational research. But research exists along a spectrum, from basic research to translational research to clinical research. Basic research is the building blocks of biological knowledge. This is where we learn biological fundamentals. So what causes the cancer to start? What's causing it to progress or metastasize? What are some new pathways to better treat the disease? Translational research is where we aim to take those discoveries from basic research and move them toward the clinic. This is where we might optimize a treatment. We'll test it in laboratory and animal models. We'll make sure it's safe and seems effective at this stage. And if it looks promising, it might move into clinical trial. And this exists in 3 phases: phase 1, phase 2, and phase 3. And each phase builds upon the next. Each phase looks at safety and efficacy of the new drug. And if by the end of the trials, the therapy is both safe and effective, it may be approved for use in humans. And so let's look at the NETRF portfolio and what our funding has looked like over the years. Since 2005, we've funded nearly $38 million in funding across 144 grants to 115 unique investigators at 74 institutions across 4 continents and 16 countries. So we are a large global funder of neuroendocrine cancer research. If you look at the pie chart, you'll see that in the blue and gray parts of the pie, about 92% of the pie is our funding of basic and translational research. A much smaller part of the pie, about 8%, is focused on clinical research that we funded. If you look at the table on the right, we also fund research across different primary sites. Because while we know that there are commonalities across disease, we know that there are also differences. And so we think it's important to fund research based on primary site. But we also think it's important to fund research based on multiple types of neuroendocrine cancer. And so that's part of our portfolio, as well as other types of research into rarer neuroendocrine cancer types, such as extrapulmonary neuroendocrine carcinomas, a very aggressive type of disease. And so how do we pick grants and how do we monitor them once they've been awarded? It first starts by issuing an RFA, which is a request for applications. This tells the scientific community that we're looking to fund grants and that we're ready to receive applications. So scientists will then send in their applications with their best ideas. We then send those applications out for a rigorous peer review process. So the applications go to scientific experts who review the applications and provide scores and comments on each application. We then meet for a peer review call where the scientific experts discuss the applications. They discuss the strengths and the weaknesses, and then they re-rank the grants at the end of the call. From there, NETRF can select the grants based on rankings as well as programmatic needs, and then they go to our full board for approval. But once the grants have been approved, our work doesn't stop there. We think it's very important to monitor the grant throughout its life cycle to make sure the grant is meeting its progress milestones. So every 6 months, grant recipients send in reports to us, letting us know of their progress. After the grant ends, we continue to receive outcomes reports, and this is a great way for us to track outcomes and impact of the work that we fund. We also have an annual research symposium every year where our grant recipients are required to attend and present their work. And so I want to end with a success story. So about a decade ago, we funded a group of investigators at the University of Pennsylvania to study CAR T-cell immunotherapy in NETs. This therapy had shown to be quite promising in blood cancers, but had been less successful in solid tumors. But we thought there was a lot of hope here, and so we gave funding to this group at Penn. So they worked very diligently to show that this has worked over time. They built a CAR T construct that worked well in mice, and this preclinical data has now allowed this work to move into clinical trial that's just started this month. And so we're very excited to see the outcomes of this work. And so with that, I want to thank our incredible research community who are in the lab every day to learn more about this disease. I want to thank our staff, our board of directors, and board of scientific advisors who make all of this possible. And of course, I want to thank you all for your time. We couldn't do this without you and your support. If you have any questions after my presentation today, feel free to follow up by the email address below. Thank you.
Hi, everyone. My name is Dawn Quelle, and I'm a professor at the University of Iowa in the Department of Neuroscience and Pharmacology. I lead the Cancer Genes and Pathways program in our Cancer Center, and I am part of a very large neuroendocrine tumor research team here, which I co-lead with Drs. Jim Howe and Yusuf Menda. And today I'm going to talk to you about discovery in neuroendocrine tumors through basic and translational research. I have no disclosures. So when we think about the journey each cancer patient navigates and the types of interventions at each stage, it ranges from preventative approaches to early detection, followed by diagnosis and specialized treatments, depending on the cancer type and stage, and ultimately palliative care and survivorship. My research primarily fits into diagnosis and treatment. What we're looking for is new biomarkers that will help define the tumor type and uncover new therapies that will best treat these tumors. So my work is both basic and translational in nature. What does that really mean, and how is that different from clinical research? Basic research is considered the foundation of medical discovery. It tells us how nature works and what causes diseases, and it's largely at the level of DNA, RNA, and protein. Now, translational research tests how those basic discoveries can be used to prevent, detect, and/or treat diseases. And it often is using preclinical models like animals. Now, in contrast, clinical research involves testing questions in people. That often will be clinical trials, but it also can be retrospective studies using tumor samples previously obtained. Importantly, we tend to think of this research continuum as a one-way kind of direction, but it isn't. The translational pathway from basic biological findings to the clinic is not unidirectional. Results from the clinic often support what we call reverse translation, where that moves science from the bedside back to the bench in an iterative process that actually helps patients faster. So in today's talk, I want to give you a flavor of this research continuum. And it's based on work from my lab where we've performed basic research using neuroendocrine tumor cell lines, translational research using mouse tumor models, and then working with clinicians to develop an early phase clinical trial in neuroendocrine tumor patients. Now, the major problem that my group is working on is to uncover new targets and therapies for NETs. Now, this is important because current drug therapies in NETs are not curative. Thus, we have a very strong need for effective therapies that will treat tumors that become resistant to the therapy. Now, as you know, NETs can arise throughout the body, but my group focuses mainly on tumors in the pancreas, lung, and small bowel. And I'm really lucky to be part of a large specialized program of research excellence, or SPORE team, at Iowa that is focused solely on neuroendocrine tumors. And our team is comprised of basic researchers like myself, as well as clinical experts like surgeons and endocrinologists and medical oncologists who treat NET patients at our very active clinic. And having both the basic and the clinical researchers working together is what really facilitates meaningful translational research. So this question that we're trying to solve right now is how to make neuroendocrine tumors and other tumors more responsive to immunotherapies, because we know that those are some of the best treatments for patients. And we focus first on pancreatic neuroendocrine tumors, or PNETs. Now, the premise of our studies is actually fourfold. The first is that clinical and translational studies have shown that anti-PD-1 immune checkpoint inhibitor therapy in clinical trials has not worked in PNET patients, despite the fact that those tumors have what we call good immune cells in the tumors, like T cells. Now, we've also done a lot of basic research, which has revealed that there are certain tumor-promoting proteins, like MEK and CDK4/6 kinases, that are overly active in PNET tumors. But monotherapies that have been tried in these patients that target CDK4 and 6 have not been effective. And then we know in other tumor types, like colon cancer, for example, or breast cancer, that MEK and CDK4/6 inhibitors promote immune cell activation, which is a good thing. And we have found in our studies that combining MEK and CDK4/6 inhibitors against PNETs actually stops their growth. So this brings me to a schematic that I hope illustrates the benefits of this combination therapy. It shows that abnormally high levels of MEK and CDK4/6 kinases, shown in yellow, cause increased PNET cell growth. And that is because they turn off a very important tumor suppressor called the RB1 protein. Now, what we and others have found is that combining inhibitors of CDK4 and 6, like the palbociclib drug, as well as mirdametinib, a MEK inhibitor, synergizes to fully reactivate the RB tumor suppressor in the tumors, stop tumor growth, and induce immune cell infiltration. So what I want to do is show you some studies that we've done in cells grown in culture as an example of our basic research showing how this combination works. Now, here we treated BON1 PNET cells with either no drug at all, either drug alone, or the combination in an assay that measures colony forming ability of the cells. Now, this combination was found to be most effective at reducing tumor cell growth as measured by fewer colonies and the size of colonies. Now, to translate that result, which was really exciting, towards patients, we moved into an animal model that was developed by our colleagues at Rutgers University. They made a MEN1-PTEN double knockout mouse, and those mice express very high levels of our target, CDK4, and those animals develop tumors by about 6 months of age. We expect to see better efficacy, including tumor regression, in the mice given the combo or the combination of drugs targeting both CDK4/6 and MEK. And this slide shows you that that prediction is true. So you can see that the combination acted synergistically to reduce that tumor size compared to either drug alone. And that's quantified in this graph, where that red bar is really small because that's the combination. And then the individual drugs are shown in the gray bars. Now, that robust level of tumor regression or shrinkage suggests that we activated anti-tumor immunity. So we quantified the tumor infiltrating immune cells in the vehicle controls versus the combination treated tumors, which are shown in red. And we found that the dual treated tumors had a much higher level of B cells and plasma cells within them compared to the controls. This was pretty darn exciting, and that's because in a lot of other human tumor types, infiltration of B and plasma cells correlates with much better patient survival. And it also correlates with the ability of those patients to respond better to immunotherapy, using checkpoint inhibitor antibodies that inhibit proteins called PD-1 or PD-L1. So if you haven't thought about this type of immunotherapy before, it is based on the fact that cancer cells express a protein on their surface called PD-L1, and that binds to a protein called PD-1 on the T cells, the green cells on the left. And that prevents your immune system from killing the cancer cells. So the use of PD-1 or PD-L1 antibody therapy blocks that interaction, and that unleashes the T cells to kill your tumor cells. And that's what we're hoping for. This led to our hypothesis that CDK4 and MEK targeted therapy will sensitize PNETs to anti-PD-L1 immunotherapy. And we tested this in the same mouse model that I introduced to you before. And we treated them with vehicle control in the open bar, with palbociclib alone in the gray bar, the anti-PD-L1 treatment alone in the red bar, and the combination in the blue. And you can see the combination caused near ablation or removal of the tumor compared to either drug alone. So in summary, we found some really exciting results here. First, that the combination of CDK4 and MEK inhibitors is effective against PNETs. And second, that we can sensitize PNETs to immune therapy by inhibiting CDK4 and 6 kinases. Now, we have a lot of future studies to do that are both basic and translational in nature, and they're listed here. But most importantly, this work has led to lots of conversations with our clinical colleagues, and we're working to develop a new clinical trial that's called a phase 0 window of opportunity trial. This is where we take patients who are already scheduled for surgical resection of their tumors, and before the surgery, we want to give them a neoadjuvant therapy of combining the CDK4/6 inhibitor with the anti-PD-L1 therapy for just 5 weeks. And then what we will do is we will compare the pre-treatment biopsies with the post-treatment resections. And we will ask if the prediction is true that this therapy will actually increase immune cells within the tumors. And we'll also take blood draws before and after treatment to see if we can monitor that immune infiltration into tumor cells by simply looking at immune cells within the blood. And we hope that this will someday lead to better treatments for neuroendocrine tumor patients. So with that, I would love to thank the people in my lab shown in the lower left, as well as my colleagues at the University of Iowa shown in the right, who are all part of our large neuroendocrine tumor spore group, and especially our colleagues at Rutgers who've done that fantastic work in that mouse model of disease. And with that, thank you very much for your attention.